Hello, we are chapter 30 and we'll be talking about green plants. In our group, we have Rini Ortega, Mark Chinchilla, Luis Gomez, Evani Goff, Claire Segura, and myself, Rosaida Hiron. In the outline, we'll be defining plants, chlorophytes and charophytes, bryophytes, trichophytes, lycophytes, pterophytes, ferns and their relatives, the evolution of seed plants, gymnosperms, plants with naked seeds, as well as what is the angiosperms. Introduction. When plants were able to establish themselves to land, it altered the history of life on Earth. This is because now the whole atmosphere could be rich in carbon dioxide and solar radiation for photosynthesis. However, the lack of water and high UV rays on land restricted green algal ancestors to an aquatic environment. Now, due to evolutionary innovations for reproduction, structural support, and prevention of water loss in plants, a larger number of alleles were then added to the gene pool, creating now a greater genetic diversity of over 300,000 plants. Plants can affect every aspect of human lives, from medicinal purposes to biofuels to building materials, food decoration, cotton for clothes. They all affect human lives differently. Defining plants. We now know that all green algae and land plant share a common ancestors over 1 billion years ago. The DNA sequence data consistently shows that a single individual gave rise to all the plants. Plants are all members of the virid plantae, including the green algae. Plants are photoautotrophic, but not all photoautotrophs are member of the virid plantae. By plant definition, it's broad, but it does exclude the red and brown algae, even though red, brown, and green share a primary endosymbiotic event 1.5 billion years ago. Plants are not fungi. Fungi, however, were essential for plants to colonize the land, enhancing the plant's nutrients uptake from the soil. In the green plant phylogeny, we can appreciate that they all share an ancestral algal. So it's not surprising that all land plants share a single common ancestor, all the way from red algae to the angiosperms. Now, the way land plants evolved from freshwater algae. A single species of freshwater green algae gave rise to the entire terrestrial lineage. Exactly what the ancestral algae is still a mystery. Close relatives, members of the charophytes, exist in fresh lakes today. The green algae was able to split into two major clades, from the chlorophytes to the charophytes. The chlorophytes never made it to land. However, the charophytes are all sisters to land plants. Land plants all share common characteristics like multicellular haploid, diploid stages, and thus diploid embryos. Land plants have had to adapt to terrestrial life. Most land plants have only limited amounts of water available. The waxy surface to prevent water loss, cuticle is one of them, as well as gas and water diffusion, which is the stomata. They also had a challenge of moving water into the plant, which increased with plant size. They had to develop specialized cells that facilitated the transport of water and minerals. This is called the tracheids. They also evolved a high efficient transport system. It's called the xylem, which conducts water, and phloem, which conducts food. Plants have adapted to terrestrial life. Terrestrial plants' exposure to high UV rays increases the chance of mutation. Diploid genome masks the effect of a single deleterious allele. All land plants have both haploid and diploid generations. They, along with multicellular green algae, have haplodiploidic life cycles that undergo mitosis after both gamete fusion and meiosis. Land plant life cycle is haplodiploidic, having multicellular haploid as well as diploid state. The haplodiploidic cycle produces alternation of generation. Red, brown, and green algae are all haplodiploidic. The diploid generation or sporophyte alternate with the haploid generation or gametophyte. Both haploid and diploid individuals can both be multicellular. Spores are also produced in meiosis. The diploid sporocytes produce four haploid spores. The spores then divide by mitosis, producing multicellular haploid gametophyte. Gametes are produced in mitosis. The gametes fuse and the zygote formed is diploid. This is the first cell of the sporophyte generation. It produces sporangia in which meiosis ultimately occurs. The relative sizes of haploid and diploid generation. The haploid generation consumes a much larger portion of the life cycle in mosses and ferns than it does in gymnosperms and angiosperms. In mosses, liverworts, and ferns, the gametophyte is photosynthetic and it's free living. The sporophyte generation can get very large. The size of the gametophyte is really limited in all plants. The gametophyte generation of mosses produces gametes at its tip. The egg is stationary and the sperm lands near the egg in a droplet of water. If mosses were at a high altitude, it would be difficult for the sperm to swim up the tree. Therefore, the small gametophyte of the sperm develops on the forest floor where gametes can meet. As mentioned earlier, when the green algae split, it gave off two distinct lineage or clades, the chlorophytes and the charophytes. The chlorophytes are special because of their unusual diversity and lines of specialization. The chlorophytes have an extensive fossil record dating back 900 million years ago. The chlorophytes can be unicellular. Early green algae probably resemble Chlamydomonas reinhardti. Microscopically, they're green, rounded with two flagella at the interior, and they're also soil dwellers that move rapidly in water by beating their flagella in opposite direction. They produce both sexually and asexually. The single-cell chlorophyte have both sexual and asexual reproduction. Unlike multicellular green plants, gamete fusion is not allowed by mitosis. Colonial chlorophytes. Colonial chlorophytes have cell specialization. It provides example of cellular specialization, an aspect of multicellularity. As in other green algae, chlorophytes store starch in their chloroplasts which contain chlorophylls A and B. Chlorophytes are extremely diverse and include both freshwater and marine representatives. Chlorophytes can also be unicellular. Chlamydomonas. Chlamydomonas is a unicellular green alga. It is a minute actively moving chlorophyte that inhabits still and freshwater pools. Chlamydomonas most often reproduce asexually. Mitosis produces as many as 16 daughter cells within the parent cell wall. Each daughter cell then secretes a cell wall that acquires a flagella. The daughter cell then escapes by secreting an enzyme that digests the parent cell wall. Chlamydomona occasionally reproduces sexually when growth conditions are unfavorable. Gametes of two different mating types come in contact and then they join to form a zygote. As seen here, they can produce both sexually and asexually. Volvox. Volvox is a well-known colonial green alga. Each cell of a Volvox colony resembles a Chlamydomona cell. A Volvox colony is a hollow sphere with thousands of cells arranged in a single layer surrounding a watery interior. In Volvox, the cells cooperate in that the flagella beat in a coordinated fashion. Some cells are specialized for reproduction, and each of these can divide asexually to form a new daughter colony. This daughter colony resides for a time within the parent colony, but then it leaves by releasing an enzyme that dissolves away a portion of the parent colony, allowing it to escape. Volvox reproductive cycles. As mentioned before, in Volvox both asexual and sexual propagation occurs. In large colonies, some kind of differentiation occurs between vegetative and non-vegetative cells, although the cells are all haploid. Here we can see the life cycle of Volvox in vegetative reproduction and sexual reproduction. The ulva. The ulva is a multicellular chlorophyte commonly called sea lettuce because it lives in the sea and has a leafy appearance. The thallus body is 2 cells thick and can be as much as a meter long. The ulva has an alteration of generation life cycles like that of land plants, except that both generations look exactly alike and the gametes as well. Ulva usually multiply by means of fragments which are accidentally produced from a thallus. Haplodiplontic life cycle. In haplodiplons, the mitosis occur in both diploid and haploid cells. Such organisms go during their life cycle through a phase in which they are multicellular and haploid, and a phase in which they are multicellular and diploid, which is the sporophyte. This phenomenon is called alternation of generation. The haplodiplontic type of cycle is found in all land plants and in many algae. Types of life cycle: diplontic, haplontic, and in plants and some algae, haplodiplontic. Charophytes. In common with other green algae and land plants, charophytes have grass-green chloroplasts containing chlorophylls A and B and store starch inside the chloroplast. Charophytes include the genus Chara. Chara is a member of the Charles and the Colocate. A member of the Coleochitales represents two clades most closely related to the land plants. Charophytes have haplontic life cycles indicating the evolution of diplontic embryo and the haplodiplontic life cycle that occurred after they moved onto land. Bryophyte. They are small non-vascular plants. It is a traditional name used to refer to all land plants that do not have a true vascular tissue. They live in moist and humid environment. They lack leaves. However, they have leaf-like scales that contain chloroplasts in which photosynthesis occur. They lack true roots. However, they anchor themselves in the soil by root-like structure called rhizoids. There are three classifications of bryophytes. These include mosses, liverworts, and hornworts. Mosses, liverworts, and hornworts. Moss. The body of the moss is usually leaf-like. It helps form the soil, colonize rocky sites, prevent soil erosion. They grow packed together in dense colonies. Many mosses have both male and female plants. Liverworts. They have many flattened body called thallus, but some have leafy appearances. The hornworts are flowerless spore-producing plant with spores typically horn-like or needle-like capsule. Here we can see hornworts, mosses, and liverworts. Hornworts develop stomata. Hornworts are one of the earliest plants. Their fossil spores date from Cretaceous period 65 to 145 million years ago when angiosperms were just emerging. The sporophytes are tiny green broom handles or horns from about 2 centimeters in diameter. It's embedded in gametophyte tissue where it gets its nutrients. However, it does have stomata to regulate the exchange of gas, and hornwort cells usually have a single large chloroplast. Tracheophytes plants, roots, stems, and leaves. Tracheophyte plants are also known for vascular plants. Cooksonia, the first known vascular plant, appeared in Silurian period about 420 million years ago, but sadly they got extinct. This plant was only about few centimeters tall. They lacked roots and leaves. They only consisted of branches, and on each tip they were homosporous. The sporangia formed at the tip of the branches. This plant, like other early plants, became successful colonizers of land by developing vascular tissue, water, and food conducting system. The vascular tissue consisted of circular or elongated cell that goes through all the plant from the tip of the roots to the edges of the leaf. The xylem, which we mentioned earlier, conducts water and dissolved minerals upward from the roots, and the phloem, which conducts sucrose and hormones throughout the plant. A cuticle and stomata are characteristics of all these vascular plants. Trochophytes include 7 extant phyla groups in 3 clades. The 3 clades of vascular plants exist today are lycophyte, club mosses, pterophytes, ferns and their relatives, and the seed plants. Whisperins and horsetail were believed to be a distinct phyla that were transitional between bryophytes and vascular plants. Now, with the phylogenetic evidence, it shows that they are closer related to the ferns and therefore grouped as pterophytes. The haplodiplontic life cycle persists, but the gametophytes have been reduced in size. Similar reduction in multicellular gametophyte has occurred as well. Stems evolved prior to roots. Early fossil records reveal stems but no roots or no leaves. Cooksonia, one of the earlier vascular plants, had transport cells in their stems but no roots limiting to the size of a plant. The roots provide structural support and transport capability. Roots are only found in trichophytes. Somewhat similar structures, though, that can transport or support non-trichophytes. The lycophytes diverged from other trichophytes before roots appeared. It appears that roots evolved at least 2 times. Leaves evolve more than once, however. Leaves increase the surface area of the sporophytes and increasing therefore their photosynthetic capacity. Lycophytes have single vascular strands that support the small leaves that are called lycophils. They may have resulted from vascular tissue penetrating small leafy protuberances on the stem. Euphils are found only in ferns and seed plants. They most likely arose from branching stems that become webbed with leaf tissue. Seeds are another innovation in some phyla. Seeds are highly resistant structure. Almost all seeds contain a supply of food for the young plant. Lycophytes and pterophytes, however, do not have seeds. Angiosperms actually adds a layer of protection to the seed and attracts animals that help in seed dispersal. This gives an advantage to the seed plant to take dominance over the terrestrial environment. Lycophytes dominate sporophytes generation and vascular tissue. They are members of 4 phyla of living vascular plants that also lack seeds. The lycophytes are relic species of the past when vascular plants evolved. They are sisters to all vascular plants. They are mostly found in the tropical areas and moist places. About 1,150 living species of club mosses superficially resemble true mosses, but once their internal vascular structure and reproductive is known, now it's clear that they are not related. Pterophytes, ferns, and their relatives. The phylogenetic relationship is among ferns is still being sorted out. They have 2 clades, that's for sure. They diverge to produce a line of ferns and of horsetails, and they're derived to yield another line of ferns. And whisker ferns as well. Whisk ferns lost their roots and leaves secondarily. They occur in tropical and subtropics. The sporophytic generation consists merely of evenly forking green stems without roots, having tiny green spirally arranged flaps of tissue, lacking veins and stomata. They are monophyletic, some taxon common ancestors with ferns. They are colorless, less than 2 millimeters in diameter and 18 millimeters long. They form a symbiotic relationship with fungi. Horsetails have jointed stems with brush-like leaves. There are 15 living species of horsetail and they are all homosporous, spores of one kind only. They constitute a single genus. They're found in swamp places. They consist of rib-joined photosynthetic stems that arise from branching underground with roots at their nodes. The stem have 2 sets of vertical tubular canals. The non-photosynthetic scale-like leaves emerge at each of their nodes. Ferns have fronds that bear sori. Ferns are the most abundant group of seedless vascular plants, with about 11,000 living species. They may be the closest relative to land plants. The fossil record states that ferns originated during the Devonian period about 350 million years ago. Here we can appreciate the life cycle of a typical fern. Here is the fern life cycle. Fern morphology. A fern sporophyte, similarly to horsetail, have rhizomes. The leaves usually develop at the tip of the rhizome as tight rolled up coils that unrolls and expands. What makes them look like a garden plant is due to plenty of fronds that are highly dissected and feathery. Fern reproduction. Ferns use both sexual and asexual reproduction methods. In sexual reproduction, a haploid spore grows into a haploid gametophyte. The sporophyte produces spores, completing the life cycle. Asexual method of reproduction include apogamy, polyphorous frond tips, and rhizome spreading. Here we can appreciate the fern reproduction, which has to do with both sexual and asexual reproduction methods. The evolution of seed plants. They came about due to evolutionary innovation, allowing ancestors of aquatic algae to colonize the harsh and very terrestrial terrains. Over the last 100 million years, both gymnosperms and angiosperms have evolved. This means that they can stop the clock and they can germinate even after a harsh winter or extreme dry season. Seed protects the embryo. The embryo is protected by a sporophyte tissue called integument. This creates the ovule. The sporangium within the ovule then divides meiotically. This then produces haploid megaspores. After this, the egg combines with the sperm to produce a zygote. The seed contains food supply for the developing embryo. A pollen grain is the male gametophyte. The functions of the gametophytes are the production of the sperm cells and the female cells and their union in fertilization. In flowering plants, the pollen grain is the male gametophyte and the embryo sac is the female gametophyte. The male gametophyte completes its early development within the anther. Conifers are the largest gymnosperm phylum. Conifers are found in the colder temperature and sometimes drier regions of the world. The most familiar gymnosperms are conifers, which are phylum Conifer Aerophyta. This includes pines. Pine morphology. Morphology is a branch of biology dealing with the study or the form of structure of organisms in their specific structural feature that can include their shape, their external morphology, etc. Pines are tough needle-like leaves that produce mostly in clusters of 2 to 5. Among the conifers, only pines have clustered leaves. The leaves have a thick cuticle and a recessed stomata due to evolutionary adaptations for retarding water loss. This is very useful since many of these trees grow in areas where the topsoil is frozen for parts of the years. This makes it difficult for roots to obtain water. The leaves have canals into which surrounding cells secretes resin which deters insects and fungal attacks. The wood in pines lacks some of the rigid cell types found in other trees, therefore it is considered a softwood. The thick bark is also an adaptation for surviving fires and low temperatures. Furthermore, some cones depend on fire to open and release seeds to reforest burned areas. Reproductive structures. All seed plants produce two types of spores, giving rise to two types of gametophytes. The male gametophyte, or the pollen grains of pines, develop from microspores which are produced in male cones. These are about 1 to 4 centimeters long and they consist of small papery scales arranged in whorls. A pair of microsporangia forms a sac within each scale. The mother cells are the microsporangia, undergo meiosis, each becoming 4 microspores. The microspores then develop into 4-celled pollen grains with a pair of air sacs, making them buoyant. A single cluster of male pine cone may produce more than 1 million pollen grains. The female pine cones, however, are produced in the upper branches of the same tree that produces the male cones. The female cones are larger and their scales become woody. In female pine cones, 2 ovules develop towards the base of each scale. Each contains a megasporangium called the nucellus. This is completely surrounded by a thick layer of cells called the integument. This has the micropyle on one end. One of the layers of the integument becomes the seed coat. A single megaspore mother cell within each megasporangium undergoes meiosis, becoming 4 megaspores. 3 of these megaspores break down and the remaining one develops into the female gametophyte. Life cycle. The male and the female gametophyte are dramatically reduced in size in these plants. Wind generally disperses the male gametophyte pollen, which produces sperm. Pollen tube growth delivers the sperm to the egg on the female cone. A Additional protection for the embryo is provided by the integument, which develops into the seed coat. Fertilization and seed formation. The female cones take 2 or more seasons to mature. While the scales are open, some of the pollen grains are trapped in the sticky fluid oozing out of the micropyle. The pollen grains being trapped are drawn down through the micropyle to the top of the nucleus. The scale closes after this occurs. When the female gametophyte is developing, a pollen tube emerges from the pollen grain beneath the micropyle and slowly digests its way through the nucellus to the argonium. During its growth, the degenerative cell divides by mitosis, with one of the resulting 2 cells dividing again. The last 2 cells function as a sperm. The germinated pollen with its 2 sperm is the mature male gametophyte. About 5 months after pollination, the pollen tube reaches an argonium and discharges its content into it. One sperm unites with the egg, forming a zygote, while all the other sperms degenerate. The zygote develops into an embryo within the seed. After dispersal and germination of the seed, the young sporophyte develops into a tree. Here we can appreciate the life cycle of a typical pine. Gnetophytes have xylem vessels. There are 3 genera of gnetophytes, phylum Gnetophyta. They are only gymnosperms with vessels in their xylem. Vessels are a common feature in the angiosperms. The members of the 3 genera differ greatly from one another. They have the Welwitschia and the Ephedra. In the Welwitschia, one of the most unusual of all plants, it's found in the desert of the southwestern Africa. The stem is shaped like a large hollow cup. It has 2 strap-shaped leaves that grow from their base. Splitting in the wind, reproductive structure has cone-like, and they produce on separate male and female plants. Now the Ephedra, they're common in the western US and Mexico, are shrubby with stems that resemble horsetails. They have tiny scales-like leaves at each node. Their both male and female structures are on the same plant. The 3 phyla of gymnosperms. Cycad, Cycas serenalis, is the only gymnosperm species found among native Sri Lankan flora. The Welwitschia mirabilis represents one of the 3 genera of the gnetophytes. Like the sperm of the cycads, those of the ginkgo have flagella. Both male and female structure in the ginkgo are produced on separate trees. In the ginkgo biloba, the only living representative of the phylum Ginkgophyta, angiosperms, the flowering plants. Angiosperms' origins are a mystery. Darwin referred to it as an abominable mystery. Flowering plants are called angiosperms because their ovules are enclosed within diploid tissue during pollination, unlike those of gymnosperms. The carpel encapsulated seeds and developed into fruits is a unique angiosperm feature. Here we can see the evolution of fruit. The leaf origins of carpels are still visible in the pea pod. Fossil of basal angiosperm. Arcobae fructus is the oldest known angiosperm in the fossil record. It is estimated to be between 122 and 145 million years. Arcobae fructus fossils have both male and female structures, but they both lack sepals and petals that evolved in later angiosperms to attract pollinators. However, it is unlikely to be the very first angiosperm. Flower house and gametophyte generation of angiosperm. Flowers. Flowers are modified stems bearing modified leaves. All share certain features regardless of their size and their shape. All flowers develop from a primordium, which is a tissue at its earliest stage of development. That develops into a bud at the end of the stalk. The pedicel then expands at the top to form a receptacle to which the remaining flower parts are formed. Flower development. Here we can see the bud of a plant and the bud that develops into a receptacle and then the remaining parts of the flower. Flower morphology. The other flower parts are attached in whorls. The outer whorl is composed of sepal. The next consists of the petals that are often colored to attract the pollinators. The third whorl consists of the stamen, which is collectively called the androsium. This is where the male gametophyte pollen is produced. At the center of the flower is the fourth whorl, known as the gynosium. This is where the female gametophyte is housed. The gynosium consists of one or more carpels. The structure of the carpel. The carpel consists of three major regions, which are the ovary, the stigma, and the style. The ovary develops into the fruit. The stigma is sticky and allows the pollen that falls on it to adhere or to stick fast. The style connects the stigma and the ovary. The style is short and may be missing in some flowers. Most species of plants attract pollinators and they reproduce. Eudicots include great majority of angiosperms. Almost all kind of trees and shrubs, peas, mints, and flowers, etc. are included here. The monocots includes the lilies, the grass, the palms, orchids, etc. They share a common ancestor with the eudicots. The angiosperm life cycle includes double fertilization. During the development of a flower bud, a single megaspore mother cell in the ovule undergoes meiosis. This is the division of a cell that results in 4 daughter cells. 3 of the megaspore soon disappears. The nucleus of the remaining megaspore division that results in 2 daughter cells, and the cell slowly expands until it reaches its original size. The female gametophyte. While the expansion of each megaspore is occurring, each of the daughter nuclei divides twice and results in 8 haploid, having a set of unpaired chromosomes nuclei arranged in 2 groups of 4. At the same time, 2 layers of the ovule differentiate and become the seed coat of a seed. 1 nuclei from each group of the 4 migrates towards the center where they function as a polar nuclei. The polar nuclei may fuse together forming a single diploid nucleus, or they may form a single cell with 2 haploid nuclei. This group closest to the micropyle is the small opening in the surface of the ovule. At the end of the 3 cells, they are called antipodals. They have no apparent function and eventually break down and disappear. Pollen production. Most anthers have patches of tissue that eventually become chambers lined with nutritive cells. The mother cell undergoes meiosis almost instantly, each producing 4 microspore. The 4 microspore at first remains together, and the nucleus of each microspore divides once in most species, and the quartet divides then separate. At the same time, a 2-wall layer is developed around each of the microspore. As the microspore contains anther continue to mature, the wall adjacent pairs of the chamber breaks down, leaving 2 larger sacs. At this point, the binucleate, which means containing 2 nuclei microscope, have become pollen grains. Pollination and the male gametophyte. Pollination is simply the transfer of pollen from its source, in this case the anther, to the receptive area, the stigma of a flowering plant. Most pollination take place between flowers of different plants through the means of wind, insects, gravity, even humans. Self-pollination takes place in 1/4 of all angiosperms. Pollination may or may not be followed by fertilization depending on the generic compatibility of the flower stigma that it has landed. If the stigma is receptive, the pollen then cytoplasm absorbs substances from the stigma and bulges through an opening. The bulge develops into a pollen tube that responds to a chemical and mechanical stimuli that guides the embryo sac. It follows a diffusion gradient and grows down through style and into the micropyle. The pollen tube usually takes several hours, up to several days to reach the micropyle. One of the pollen two cell lags behind, and its nucleus divides into pollen grain or in the pollen tube, producing two sperm cells. At this point, the pollen grain along with its tube and sperm has become a mature male gametophyte. Double fertilization and seed production. As the pollen tube enters the embryo sac, it destroys the synergrin in the process and discharges its content. Both sperm and functionable leads to double fertilization. The first sperm unites with the egg and forms a zygote, which develops into an embryo. The other sperm and the two polar nuclei unite, forming a triploid primary endosperm nucleus. Double fertilization and seed production. The primary endosperm then begins to divide rapidly and repeatedly, becoming a triploid endosperm tissue that may soon consist of thousands of cells. Endosperm tissue can become then an extensive part of the seed in grasses such as corn, and it provides nutrients for the embryo in most flowering plant. Thank you.